Hello, in this video we're going to focus on lung physiology, focusing on gases and pH. And we'll also look at the oxygen dissociation curve. So air enters the respiratory system in the upper airways, proceeds to the conducting airways, and then onto the respiratory airway made up of the branches of the bronchi, bronchioles, terminal bronchioles, and finally ends in the alveoli. You can think of the alveolus, which is singular, of alveoli as the building blocks of the lungs where gas exchange occurs, gas exchange occurring between the lungs and the bloodstream. The gas exchange in this case is between oxygen and carbon dioxide, or CO2. So just recapping, the basic physiology: the heart will pump deoxygenated blood from the right ventricle towards the lungs. Deoxygenated blood means blood containing a higher concentration of CO2 with lower amounts of oxygen. In each alveolus, the carbon dioxide will be dropped off and exhaled out of the body, so removing carbon dioxide from our body. And then oxygen will be inhaled and will then reoxygenate the blood. This newly oxygenated blood, or reoxygenated blood, will then go back to the left side of the heart. The heart will then pump this oxygenated blood around the body to the other organs, such as the brain. The oxygenated blood delivers oxygen to organs, tissues. The organ tissues then uses up this oxygen to help create energy in the form of ATP. Now, as a process of creating this energy, ATP, the organs, the tissues will form a byproduct, which is carbon dioxide. Carbon dioxide is then made and is actually expelled into the bloodstream as exchange, you can say. Now, because carbon dioxide is in the bloodstream, the blood is now deoxygenated once again, and this deoxygenated blood will return to the right side of the heart. The right side of the heart will continue the cycle. It will pump blood to the lungs to reoxygenate the blood. Oxygen itself travels in the body mainly in red blood cells. Oxygen binds to these molecules called hemoglobins, and we have millions of these in each red blood cell. In deoxygenated blood, with deoxygenated hemoglobin, or deoxyhemoglobin, the structure of hemoglobin here is referred to as a tight structure. Hemoglobin, just recapping, is a protein made up of 4 subunits, each of which contain a heme moiety attached to a globin chain. And each globin chain contains iron, where oxygen normally binds to. Each of the 4 iron atoms in hemoglobin can reversibly bind one oxygen molecule. Let's see what happens when deoxyhemoglobin receives oxygen. Well, the reaction is rapid, requiring less than 0.01 seconds. Remember, in deoxyhemoglobin, the globin units are tightly bound in a tense configuration, in a T configuration, which means that it actually reduces the affinity of the molecule for oxygen. When oxygen is first bound, the bonds holding the globin units are released, producing a relaxed configuration, or an R configuration, which exposes more oxygen binding sites. So to put it simply, you can imagine it like a cascade. Firstly, the hemoglobin is in a tense configuration with no oxygen. And then when oxygen starts to bind, it will become more relaxed, a relaxed configuration, which actually increases the affinity for oxygen even more. Kind of weird. This whole thing is called positive cooperativity. So by the end, the hemoglobin molecule is an oxyhemoglobin because it is filled with oxygen and it is in a relaxed form. So from the lungs, you now have heaps of oxyhemoglobins, which means your blood is reoxygenated really. And this reoxygenated blood is ready to be delivered to the body tissues. It's a good idea now to briefly talk about partial pressures of oxygen and carbon dioxide in deoxygenated blood and also in oxygenated blood. because it will be relevant to what we'll talk about later. Now you can imagine that when blood is oxygenated or reoxygenated from the lungs, it would have a higher partial pressure of oxygen. So higher amounts of oxygen, 100. And carbon dioxide is low, 40. The oxygenated blood I'm talking about are found in your arteries, such as your aorta. Deoxygenated blood, on the other hand, is very different. When we talk about deoxygenated blood, we are talking about the veins. So oxygen, it can be up to 40 millimeters mercury, and carbon dioxide about 45. The changes in partial pressure is important to understand because it helps explain the hemoglobin dissociation curve, and we will talk about that now. The hemoglobin dissociation curve, or the oxygen hemoglobin dissociation curve, relates a percentage saturation of the oxygen carrying power of hemoglobin to the partial pressure of oxygen. Now that might sound crazy, but let's just try to understand it together. So on the x-axis, you have the partial pressure of oxygen, 25 to 100 millimeters mercury. And on the y-axis, you have the saturation of hemoglobin, which what we're looking at specifically is oxyhemoglobin in percentage. This saturation of oxyhemoglobin means how much oxygen is bound to the hemoglobin basically. The more oxygen bound to hemoglobin, the higher the oxyhemoglobin, and therefore the higher the saturation. So the oxygen hemoglobin dissociation curve has a characteristic sigmoid shape due to the tense and relaxed configuration interconversion. There is a plateau here where the partial pressure of oxygen is nearly high. The plateau signifies that high partial pressure of oxygen does not cause large changes in oxygen saturation of hemoglobin. So at the plateau, you can see that the partial pressure of oxygen of 100 already has an oxygen saturation of 98%. So there's not much change. Whereas before the plateau, so a partial pressure of oxygen of 50, let's just say, already has an oxygen saturation of 85%. So a dramatic change. So note that small changes at low partial oxygen pressure leads to large changes in oxygen saturation. Another example is that a low partial pressure of oxygen, such as 26, has already oxygen saturation of 50%. The reason for this sigmoid shape and why low partial pressure of oxygen has a quick increase in oxygen saturation is because of positive cooperativity. When the first heme in the hemoglobin molecule receives oxygen, the affinity increases for the second and then the third and so on. Now let's look at factors which can shift this curve, which are essentially factors affecting the affinity of hemoglobin for oxygen. Three important conditions affect the oxygen hemoglobin dissociation curve. These are the pH, the temperature, and the concentration of 2,3-diphosphoglycerate, or DPG. Here is the curve normally. We will look at examples of what shifts the curve to the right and what shifts the curve to the left. When the curve shifts to the right, it means that there is reduced hemoglobin oxygen affinity, and this occurs in tissues. So basically, the hemoglobin affinity for oxygen is decreased. Remember the normal curve, the partial pressure of oxygen of 50 had an oxygen saturation of about 83 to 85%. In this new curve, the partial pressure of oxygen of 50 has an oxygen saturation of 70%. So reduced oxygen saturation, and this is because there is reduced affinity for oxygen. Things that cause this to happen include an increase in carbon dioxide, which really means a decrease in pH. Other things that causes the shift to happen include an increase in temperature and an increase in DPG or BPG. It's good to remember that the reduced hemoglobin oxygen affinity occurs in tissues in general. Because when you think about it, you want to give your oxygen to the body tissues so that they can use it. These tissues include the placenta because you want to offload oxygen to the fetus, and muscles cells, especially during exercise. Now the curve can also shift to the left here in red. What happens here is that we see higher hemoglobin oxygen affinity. And for this example, you can think of the lungs because it is in the lungs where oxygen comes and binds to hemoglobin. And so you would expect to see higher hemoglobin oxygen affinity here. Later, when the blood travels to the body tissues such as the muscles, the affinity reduces, as we have learned. The other factors which contribute to the high hemoglobin oxygen affinity, so this red curve, are reduced carbon dioxide, which means higher pH, reduced temperature, and also reduced DPG, which is essentially opposite to the curve when it shifts to the right. So let's compare now. In this red curve, so a high hemoglobin oxygen affinity, if the person has a partial pressure of oxygen of 50, the oxygen saturation is already 90%. So in this video, we talked about how oxygen binds onto hemoglobin from a deoxygenated state in a tense configuration into a relaxed R configuration. And we also talked about the oxygen dissociation curve and factors which cause the curve to shift to the right, as well as factors which cause the curve to shift to the left. Hope it made sense. Thank you for watching.